0610_W23_Ms_42. 1a. Red blood cells are specialized cells that transport oxygen. State the substance in red blood cells that combines with oxygen. Hemoglobin. B. State the name of the component of blood that promotes blood clotting. Platelets. C. Some students investigated the effect of immersing red blood cells in different concentrations of salt solution. They measured the diameters of samples of red blood cells and calculated the mean. They then immersed each red blood cell sample in a different concentration of salt solution. After 2 minutes they measured and calculated the mean of the samples again. Table 1.1 shows the results. I. Calculate the percentage increase in the mean diameter of red blood cells that were immersed in the 0.8% salt solution. Give your answer to two significant figures. 9.3. 3. MP1 selection of correct values from table. MP2 correct calculation to any number of significant figures. MP3 correct rounding to two significant figures. E. Explain the results for the red blood cells that were immersed in the 1.8% salt solution. Any 3 from. Salt solution has a lower water potential than the red blood cells. Diameter decreases because water leaves the red blood cells by osmosis. Water travels from higher water potential to lower water potential down a water potential gradient. Across the cell partially permeable membrane. E. State why there was no change in the mean diameter of the red blood cells immersed in the 0.9% salt solution. Any 1 from. Water potential of red blood cells is the same as the salt solution. No. Net overall movement of water across membrane, in or out of cell. D. State why red blood cells burst when immersed in pure water but plant cells do not. Red blood cells do not have a rigid cell wall. E. State 2 uses of water in a plant. Any 2 from. Transport of ions, sucrose, translocation. As a reactant, used in photosynthesis. As a solvent, substances dissolve in it. Medium 4. Chemical, enzyme, metabolic, reactions. Support, maintaining turgidity of cells, maintaining turgor pressure, prevent wilting. E.g. cooling, temperature regulation. Germination, elongation of cells in growth. 0610_S23_Ms_41. 2a. A student investigated osmosis in potato plant cells. He immersed cubes of potato tissue in water and different concentrations of sucrose solution. For 30 minutes. The masses of the potato cubes were measured before and after immersion. The percentage changes in mass were calculated. Table 2.1 shows the results. I. Using the information in Table 2.1, calculate the percentage change in mass at 1.00 moles dm3. Give your answer to 2 decimal places. Minus 13.28%, 3. MP1 for correct selection of data from Table 2.1. Equals 1.111.28 or minus 0.17. MP2 correct calculation minus 0.17 or 1.11. 1. 28 over 1.28 100 or minus 13.28125. MP3 answer rounded correctly to 2 decimal places with a minus sign. E. Using the information in Table 2.1, explain the difference in the results between the 0.6 moles dm3 and the 0.8 moles dm3 sucrose solutions. Use the term water potential in your answer. Any 5 from. Potato cube in 0.8 moles dm3 solution loses greater percentage mass. Movement of water out, loss of water, is cause of mass loss. Water moves from an area of high water potential to an area of low water potential. Water potential of 0.8 moles dm3 solution is lower than the water potential of the 0.6 moles dm3 solution. Greater, steeper, water potential gradient in 0.8 moles dm3 than in 0.6 mm dm3. Relatively more water leaves the potato cube in 0.8 moles dm3 solution. E. Describe the expected appearance of a cell from a potato cube that has been immersed in distilled water for 30 minutes. Any 2 from. Cell is swollen, larger, bigger, wider. Semicolon. Cell is turgid. Vacuole is swollen, larger, bigger, wider. Cell wall bulges. Cell membrane, cytoplasm, cell contents, presses on cell wall. B. Describe how the process of active transport differs from the process of osmosis. Any 3 from. Uses energy from respiration, mitochondria. Transport movement is against a concentration gradient. Involves movement of sugars, ions, substances other than water. Involves protein carriers. C. State the type of plant cells that use active transport to absorb mineral ions from the environment. Root hair cells. D. Explain the effect of a lack of magnesium ions on the color of plant leaves. Leaves are yellow, leaves show chlorosis. Magnesium required for making chlorophyll. 0610-S23_Ms_43. 8. The movement of molecules within an organism can occur by diffusion and active transport. Complete Table 1.1 by placing ticks 3 to show the correct features of each process. Feature active transport. Diffusion movement of particles always occurs across a cell membrane. Molecules moving during gas exchange. Rate of movement of particles is higher when the concentration gradient is larger. Requires energy from respiration. B. Explain why active transport is important in root hair cells. Any 3 from. For ion, uptake, absorption. Ion concentration lower in the soil than in the root cell. Movement against the concentration gradient. Creates a water potential gradient. Named example of ion e.g. nitrate, magnesium. 0610-S20_Ms_41. 3. 8. Dialysis tubing is an artificial membrane, which is similar to the lining of the intestine. A student investigated the diffusion of glucose through dialysis tubing by using the apparatus shown in Fig. 3.1. The student took samples of the water outside the dialysis tubing at 5-minute intervals and tested the samples with Benedict's solution. The results are shown in Table 3.1. I. Describe and explain the results shown in Table 3.1. 3. Any 3 from. Blue at time 0 indicates no glucose present. Ensures that no glucose on outer surface of dialysis tubing, in water, as a result of an error. Green, yellow, red, indicates presence of glucose. Glucose diffuses, moves, out of dialysis tubing, into water. Movement is down the concentration gradient, high to low concentration. Dialysis tubing is permeable to glucose. E. The student repeated the investigation with a higher concentration of glucose in the dialysis tubing. Predict the results that the student would observe. Idea that Benedict's solution changes color quicker, gives more intense color. B. Fig. 3.2 shows a drawing of a cell from the lining of the small intestine. The lumen is the space inside the intestine where food is digested. State the names of the 3 labeled structures in Fig. 3.2 and describe the role of each structure in the intestinal cell. Are microvilli. Function. Allow movement of substances into the cell. Increase surface area for absorption by diffusion or active transport. Have proteins in the membrane for active transport. B is the rough endoplasmic reticulum, RER. Function. Site of protein synthesis. Modify proteins. Assemble amino acids in a specific sequence to make name protein. C is a mitochondrion. Function. Aerobic respiration. Provides energy for named cell processes. C. The cholera bacterium can survive in the small intestine and the large intestine. The bacterium releases a toxin that interacts with receptors on the surface of cells. Fig. 3.3 shows the effect of the toxin. The arrows indicate the direction of movement. The toxin stimulates the secretion of ion X out of the intestinal cell. I. State the name of ion X. Chloride. E. Describe the effects on the body of the secretion of ion X into the lumen of the intestine. Any 4 from. Loss of water by osmosis, down water potential gradient, diarrhea, dehydration, loss of other named ions, salts.